Hello everyone, welcome to VLearn and welcome back to the course Introduction to the Genetics. I am Ashish Kumar and in this class we are continuing the Introduction to the Genetics part. In the last class we studied about Mendel experimentation. So if we talk about Mendel experimentation, we have already explained that the Mendel experimentation was basically divided in 3 steps. So the hybridization experiment performed by Mendel, this is known as hybridization experiment, and this hybridization experiment performed by Mendel, so it's Mendel experimentation, Mendel experimentation. This Mendel experimentation or hybridization experimentation take place in 3 steps. So in this class, we are actually going to study how Mendel performed these 3-step experiments on the Pisum sativum. The first step was selection of the parental plant. As we discussed in last class, we know that these parental plants were selected by pure line selection method. What do you mean by pure line selection method? Pure line selection is basically the plants or the progenies obtained by continuous self-pollination or self-fertilization. So these parents plants are basically obtained by pure line selection method. So Mendel took selected two plants by pure line selection. One was tall and one was of course dwarf. So this is the first step, that is selection of the parent plant. Selection of the parent plant. The tall plant was a female plant. If we talk about the female plant was tall plant, whereas the dwarf plant was a male plant. He crossed between two of these. By crossing these plants, of course, what he did was cross pollination. This is strictly cross pollination, and for ensuring this strict cross pollination, what he did was he did emasculation. Emasculation is basically removal of the anther part from the flower. The second thing he did was he covered the flowers with a bag. So this process is known as bagging. Bagging is basically for preventing unwanted fertilization, unwanted cross pollination. So it's basically for preventing cross pollination. The third step was dusting of the pollen grain, that is dusting of the male part, that is anther gives you the pollen grains, and these pollen grains were spread from the male part to the female part, and this process is known as dusting. Dusting or dusting of the pollen grain. The fourth step was to rebag. Rebag is again to ensure or prevent any type of unwanted cross pollination. So this is cross pollination. The seeds from this cross was obtained, and these seeds were collected, and these collected seeds were sown in the next season, sown in the field for the next season. So what he did was he took the selected the parented plant and he did cross pollination. So first part is selection, and second part is what? Cross pollination. So two parts are done. In cross-pollination, he did emasculation, bagging, dusting, rebagging, and he obtained these seeds. These seeds were sown in the field for the next season, and the results were observed. He observed that all the plants obtained by growing these seeds into the field were tall. Or you can say the 100% of the plant were tall plants. That is, the progeny obtained from this tall plant and this dwarf plant, we obtained all the tall plants. No dwarf plant was observed in the first progeny. Then these first progenies were again collected by the Mendel. So he collected these seeds, he collected these plants and collected seeds from the, he collected these plants and he did self-fertilization, restricted self-fertilization. So in the next stage, the progeny which were obtained in the last stage, these progeny were self-crossed, that is self-fertilization. Upon self-fertilization, after self-fertilization, seeds from these plants were again obtained. This is the second generation. This was the first progeny, that is first generation. This was the first generation. The first generation from parental plant. These seeds were collected and sown into the ground for the next season. Sown for next season. And these seeds and results were collected. What he observed was, he observed mixed variety of plant. In the first progeny, all plants were tall plants. All plants were tall plants. Whereas in this progeny, that is the second progeny, second generation, the second generation was a mix of tall plants and dwarf plants. But if we talk about number of tall plant, the ratio of the tall plant to the dwarf plant obtained by Mendel was 3:1. Or you can say is 75% of the plant in the second generation were tall plants, whereas 25% of the population was dwarf plant. So these were the results obtained by Mendel. This was the cross with respect to only one character. He performed cross for all the characters and similar results were obtained for all the 7 varieties of character, all the 7 pair of character. So this was Mendel experimentation part. This was performed in 3 steps. The first step was selection of the parental plant. The second stage was the cross-pollination, and third stage was self-pollination or self-fertilization. Upon self-fertilization, he obtained the ratio of 3:1, that is 75% to 25%. This is how Mendel observed the 3:1 ratio in the Pisum and Sativum. In the next class, we will discuss how this ratio gave rise to the principles of this inheritance. So based on these experimentation part, Mendel gave principles of inheritance. So in the next class, we will discuss the principle of inheritance. Thanks for watching the video. Hope you liked and enjoyed the video. Please don't forget to give your feedback on each video, and please don't forget to hit like, comment and subscribe button. Thank you very much.